Your platform uses enzymes to create new types of molecules. Can you explain that in a simple terms?
Peptides are really a hot space right now. And a peptide is, in its simplest terms,
There are many diseases still don't have effective treatments. How can Sethera help address those challenges?
a lot of diseases are difficult to treat because they're biological targets that are hard to drug.
Building the company is hard. What has been the biggest challenge in building Sethera from the ground up?
So it's a really complex there's lots of dimensions here.
Correct. Right.
So
Welcome to Life Science Insights podcast. I'm your host Sandil Karnanidhi. Today, I'm excited to welcome Karsten Eastman, CEO and co-founder of Sethera Therapeutics, a company pioneering enzymatically constrained peptides to unlock new possibilities in drug discoveries and therapeutics. In this conversation, we'll explore Karsten's journey, the science behind Sethera's platform, how enzymes can be used to create entirely new classes of therapeutic molecules, what this could mean for the future of treating diseases. Thank you for joining us.
thank you so much for having me here, Sen. I'm really excited for the conversation. So looking forward to it.
Can you start by telling us a bit about your background?
Of course. So my background is in chemistry, specifically enzymology and the study of enzymes and how they relate to processes in nature. I did my PhD at the University of Utah, where I was looking at some very unusual enzymes that modify peptides. And I really became fascinated with this idea that nature's evolved these incredibly complex and very precise molecular machines for making unique complex chemical structures.
What inspired you to start Sethera Therapeutics?
So the inspiration for Sethera was realizing that these enzymes could be used not just to study biology, but to actually make entirely new classes of drug-like molecules. We saw an opportunity to take this neat enzymatic chemistry that mostly lived in academic labs at that point and turn it into a true drug discovery engine. And I really wanted to translate that deep chemistry into therapeutic impact. I know that there are a lot of diseases that are important, but we still don't have good ways to drug them. And Sethera was founded to help close some of that gap.
What problem in drug discovery were you trying to solve when you founded the company?
The big problem is that there are many disease-driving proteins that are not very easily addressed by traditional drug modalities. Everyone is very familiar with small molecules, which are very powerful as therapeutic entities, but they often struggle with the flat or dynamic protein surfaces or protein-protein interactions, where you have, the proteins that are rubbing up against each other to cause some sort of downstream effect. On the other side, you have antibodies, which are also quite powerful as therapeutic entities, but they are not very penetrant, so you can't reach those intracellular targets. We wanted to build a platform that can blend the best of both the hyperspecificity of antibodies and also the best properties of small molecules like oral bioavailability, penetration, and that's where the Sethera platform started to evolve from, trying to blend the best properties of small molecules and biologics in the world of peptides.
For people hearing about Sethera for the first time, what does company do?
So we discover new peptide-based medicines using enzymes. I think that that's the simplest way to think about it. We start with a very, very large library of peptide sequences. We then use enzymes to transform those massive libraries into structured drug-like molecules, and then we screen them against very diverse biology in order to find activators, deactivators, or binders that may become therapeutic leads. The goal is to make molecules that can address diverse and difficult targets, especially where those conventional approaches I mentioned before have struggled.
What makes Sethera different from other biotech companies developing new medicines?
Our big selling point here is that the entire platform is built around enzymatic chemistry.
Okay.
We're not just screening ordinary peptides
Hmm.
or small molecules or even biologics. We're using these enzymes to make these unique peptide architectures. And that really sets us apart. We can access completely different chemical space, and the molecules that we make are very structured, very diverse, and built to engage difficult targets.
Okay, that's a differentiator, right? Your platform uses enzymes to create new types of molecules. Can you explain that in a simple terms?
Sure. So peptides are really a hot space right now. And a peptide is, in its simplest terms, a chain of individual amino acids. And it's the identity of those amino acids that gives the peptide its property.
Mm,
However, a peptide is just a small protein. And our body is very good at chopping those small proteins down and back into the individual bits. Our enzymes act basically like you could think of a molecular stapler. We take the peptides, staple them together in different shapes and different forms,
mm,
and that gives it a defined shape. And that defined shape is often something that has never appeared before in nature.
okay.
And because of that, it makes it so it can bind more specifically, resist degradation, and behave more like a medicine.
Why are enzymes such a powerful tool for discovering new medicines?
So they're very selective and efficient. One of the complexities about making a peptide therapeutic is that peptides often have repeating handles or repeating units that
Okay.
makes it very difficult to selectively build a bond into. And for drug discovery, that matters because we want to make really large numbers of complex molecules
Mm,
in a very repeatable way that's compatible with screening.
mm.
And enzymes allow us to build that chemical complexity directly into the libraries. What this means is we can functionally explore shapes and architectures that would otherwise be slow, difficult, expensive, or in some cases, impossible to access, but we can access them.
What are enzymatically constrained peptides and why are they exciting for drug discovery?
Enzymatically constrained peptides are, as the name suggests, peptides that have been linked or locked together into certain shapes using enzymes. This is really important because Biology 101 teaches us that structure equals function. And there's lots of other peptide companies that are using macrocycles or constrained bicycles as part of their search, which is great. That's a really strong foundation for peptides.
Mm,
But one of the limitations there is if all you can make is a macrocycle, which is just a circle, you can have all of this chemical diversity on the outside of the circle, which is
okay.
great.
Mm,
But that means you're limited to one architecture.
okay.
And Sethera is not limited to the one architecture. The enzymatically constrained peptides
Mm.
means that we can address targets that are far more complex.
There are many diseases still don't have effective treatments. How can Cetera help address those challenges?
So a lot of diseases are difficult to treat because they're biological targets that are hard to drug. It could be that the target is intracellular and small molecules can't address it. Sometimes it's a big protein-protein interaction and the binding surface is large or shallow or structurally complex.
Okay.
We are building molecules that can go after exactly those types of targets. The big goal is to use the architectures that we're generating to open new therapeutic possibilities. And chemistry just hasn't been able to deliver the right kind of molecule yet, and we believe we can.
What kinds of drug targets are difficult to reach with the current technologies?
Uh, so protein-protein interactions, that's a good one that I've mentioned a few
Yes.
times, but there's
Yeah.
other things like, say, transcription factors, intracellular signaling proteins.
Okay.
A big one is mutant proteins that drive cancer, where you don't want to target the normal protein, you only want to target the mutant. So you need to have
Mutants.
something that's very specific.
Okay.
Or there's certain extracellular targets where antibodies are too big to really address. They don't have a lot of these targets don't have obvious pockets for small molecules to drug. And it's this piece where there's often really clear biology, but we don't have the right molecule or the right molecular tool to address it quite yet.
How does your platform help discover therapies for these hard to treat targets?
So the biggest piece is the architectures.
Okay.
We don't just vary
Mm,
the identity of the amino acids in question. We vary the architectures,
mm,
which means that rather than being stuck with just a circle, which is a macrocycle, we are a polymacrocyclic peptide platform, which
mm.
means that we can make and layer on additional higher order architectures. And that's how the platform directly discovers therapies, is we're not just building single architectures, we're building many architectures from the initiation of the screens.
Okay. Since you're in the innovation and data space, I have few topics to discuss on this. How do you find the test new molecules on your platform?
What we do is we basically have a library that's barcoded.
Mm.
And the nice thing is that, the big question is, how do you find the golden needle in the haystack? How do you
Okay.
build build the magnet to pull
Uh,
out the golden needle in the haystack, right?
okay.
We take a peptide. That peptide is bound or connected to a barcode, which
Mm.
is its encoding sequence. We then can make more than trillions of those peptides. All of them are enzymatically constrained. We then run them over the biology of interest and wash away everything that doesn't bind. What we
Mm.
can then do is amplify the signal of the barcode and then
Okay.
start the process over. So we'll do rounds of enrichment to make sure that the signal that we're receiving from the biology of interest is
Mm.
accurate. We'll sequence everything that comes out, then resynthesize the peptides that show up the most frequently from those rounds of screening.
And data is a new oil. What
Yep.
role does data play in helping you make better drug discovery decisions?
Yeah, so every screen gives us information not only
Yeah.
about the individual peptide sequences, but there's broader patterns that we see typically at play too. Which architectures perform the best, what motifs are enriched, because we
Mm.
see not only amino acid enrichment, but also architectural enrichment, and
Mm.
then how different target classes respond to different molecular shapes.
Mm.
That's one interesting thing that we're seeing is that it's not just enrichment of amino acids, it's enrichment of architecture. And that will really allow us to make better decisions over time. We learn that, say, certain library designs are more productive and just work better with certain targets. And it's really a strategic asset in the long run, too. Every screen improves our understanding of how these constrained peptide structures relate to both target binding and therapeutic potential.
And AI is a hot topic. Are you
Yep.
using AI or machine learning? How do they
So
support
that's
your work?
Yeah, we're in the early stage here. We're
Okay.
building very large data sets
Okay.
that can then be translated to better AI and better ML. I
Okay.
think that it's really a question of, if you have good data, you'll end up having a great AI, and if you have bad data, you'll get something that's not very productive.
Correct. That's
So
true.
we're still in the process of building a strong foundation of our data to then support AI and ML.
ML.
And that means that as we build more validated data sets,
Mm.
the AI will then help us build better libraries, prioritize candidates, and reduce the number of molecules that we need to test experimentally. But one piece that sets Sethera apart is that we already are building in real space and real time all of these molecules, which means that in some cases we're actually ahead or upstream of AI, but
Mm.
we can still use our data to make better AI in the future.
Building the company is hard. What has been the biggest challenge in building Cetera from the ground up?
So it's a really complex there's lots of dimensions here.
Correct. Right.
So in academics, you can really focus on giving more evidence, on trying to prove something that's scientifically interesting, or maybe sometimes isn't scientifically interesting, but it gives you grant dollars.
Mm.
Um, but at a company, there's other pieces that you have to do. Can it be repeated? But can it scale? That's a big question. Scaling.
Mm.
Uh, can it actually produce something that has commercializable worth? Can you protect it, or can someone just steal it? And can you then explain it clearly to potential investors, partners, patients? And
Mm.
that's hard. You have to tr take these scientific breakthroughs to like transition them, build them, and
Correct.
that is a big challenge.
Big challenge. And moreover, building a team is also a big challenge, right?
It is, yeah. Finding the right people getting one thing I have to say, Sethera has fantastic people, and I'm very grateful to them. I'm working with a lovely team right now, so I'm glad to have them.
How do you balance cutting edge science with the running a growing business?
I try to keep both the scientific mission, but also the business connected. If your science isn't ambitious, then uh, it's hard. It has to be ambitious, and we're combining a lot of pieces that I don't think that have ever tried to be combined before. At the same time, the company still has to make decisions based on milestones, validation, and focus. Since we're a platform, that focus question has been one of the hardest things to answer. We have so many doors we can walk down. How do we prune off the doors that might be less productive and focus our attention on the doors that will give the highest value? That means there's questions we have to ask of stuff like, hey, what experiment is going to reduce most of the risk? Or what would make this program more valuable and more actionable? Or
Okay.
what would a partner want to see from this? And that's it's not enough for it to be scientifically exciting. It really has to be moving the company towards those milestones, those validations. And that's what we try to do, keep the science interesting, but also build something that has value and meaning.
What qualities do you look for when building your team?
Yeah, so I'm glad you transitioned into this. I mentioned that I'm grateful for the team. It's not just people who are technically excellent. There's so many people out there who are just very good at doing specific work, but sometimes lack a little bit of adaptability. And that's one of the things that think is very important. For an
Mm,
early stage company, things can move very quickly, and people have to be able to be solve problems rapidly, and many of those problems don't have a predefined playbook or an instruction manual. It's one thing to be able to follow a protocol. It's something else when you have a problem that has never existed before. And that's a lot of what we look for. I do value scientific honesty as well. Drug
okay.
discovery is hard. It
Yeah.
is. And you need people who can look at the data and tell you what's working and be very clear about what's not. And
That's right.
that's one piece that my team has been very good at doing. So, people that are creative, collaborative, and think that, well, the science and what you're building is neat. You don't want your team to be bored either.
Yeah. And more importantly, what not, right?
Yes, exactly.
What trends in biotech are you most excited about right now?
So it's really hard to pick, actually. The whole data-driven discovery is nice. I think that it'd be nicer if there were a few more wins in that space. There's been a lot of hype around it, and I'm not saying that the hype isn't well-founded. It's just that it hasn't quite delivered as much as I think people expected initially. I still think that it's a very neat trend. But then, new modalities or new ways of combining multiple ideas into one space and better screening technologies. And I think that Sethera definitely sits in the better screening technology
In
space.
technology, right?
Um, drug discovery for a long time was dominated by small molecules and antibodies.
Mm,
They're still important, but I'm excited to see other modalities hopefully become just as important, or if not better in the future.
better in the future. Yes. How do you see peptide-based medicines changing healthcare in the future?
Right now they're having a resurgence,
Mm,
and I mean, all the GLP-1s have made that very clear. But yeah, it's peptides are having a big impact, right?
okay.
Metabolic disease, oncology, endocrinology, and even skincare. And yeah, I mean, I'm sure you've seen some of the influencers on the various, TikTok, Instagram, Twitch, YouTube have all been talking about, peptide supplements. I don't support those, but I'm saying that, there's a trend of peptides becoming more popular. And I think that as we get better at controlling peptide structure, stability, delivery, making it so that they're more orally bioavailable options, peptides can address a lot that small molecules and antibodies can't. So there's going to be a lot more precision that peptides enable.
Where do you think the drug discovery will be 5 to 10 years from now?
I think there's going to be a lot of integration. We're seeing the early evidence of that where you can take screening, you can take structural biology,
Mm,
a lot of the high throughput chemistry, and then an AI layer on top of it, and all of those will be bound quite tight.
Mm.
And it's platforms will be able to have this continuous learning layer from each experiment where the data and the information from it can continually feed back and feed into the next round of design. And there's going to be emphasis on treating things that were previously considered undruggable, and more targets will become accessible, more biology and biological manipulation will be in the realm of possibility. So there's drugged human health will change a lot in the next 5 to 10 years. Right now, that there are so many options, and that you've built these peptides that can interact with not just one, not just two, but
Mm.
even three targets or more simultaneously.
Okay.
And that's one of the neat levels or areas that sets peptides apart from small molecules and is a unique differentiator too, even from biologics. You
Okay.
hear about these, bispecific antibodies or trispecific antibodies.
Okay.
Well, peptides are achieving quad specifics. So
Mm.
that it means that one therapeutic entity, one molecule
Mm.
can treat not just one thing. So for the future of drug discovery, there's this modular aspect of peptides that will allow for combination therapies, but rather than needing to take two separate drugs for it, you just have to take one,
One.
and it ends
Okay.
up being a very, very potent, useful, and beneficial drug for humanity. And the GLP-1s, I think, are the first, not necessarily the first true example, because bispecific antibodies were a thing before this,
Mm.
but the first very well accepted and very well commercialized example that everyone recognizes.
What is your long-term vision for Cetera, and what impact do you hope it will have on patients?
Mhm. The long-term vision is to have Sethera become a company that repeatedly discovers new and useful medicines against targets that were either difficult or completely inaccessible. I don't want Sethera to be defined by a single molecule or a single program. I don't want that at all. The platform is truly designed to be repeatable, and that turns into the ambition to build a pipeline across broad therapeutic areas where the peptides that we're making can make a real difference. I hope to make medicines and for patients who don't have good options. If we can take targets that are biologically compelling but historically hard to drug and use our platform to make a real therapeutic program, that would be a meaningful contribution. I don't want to do it just once. I want to
Mm.
do it multiple times across a lot of very difficult to treat biology.
It's been a pleasure having you on the show. Thank you for sharing your story, your expertise, and the exciting work being done at Cetera Therapeutics. This was an incredible, insightful conversation, and we're excited to see how technology continues to expand what's possible in medicine. We appreciate your time and look forward to follow your journey.